morning, everyone. I'm Joni, and I'm really happy to introduce both Sam and Adriana for today's joint seminar. Sam and Adriana are both members of our relatively newly minted research group, the Northern Shark and Ray Research Group, which is built from Peter Kine's legacy here at CDU, and now I'm taking the reins of. So really excited to showcase some of the work that our students are doing, which is great. So Sam's an honours student, going to present on some of his honours research, which is part of a long-standing collaboration with the Malik Ranger Group on the Daly River. And Sam's also a research assistant in our lab as well. And Adriana is going to talk about her important shark and ray areas research and the broader sort of more international focus as well. So keep it brief. These presentations too are going to be given at the Indo-Pacific Fish Conference in Taipei in June. And so, yeah, any feedback that you have on these presentations, I'm sure would be welcomed. And yeah, leave it up to Sam to take it away.
Hi, everyone. My name's Sam, for those of you who don't know me. So today I'm going to be talking not just about the work that I've done for my honours, but also more broadly about the collaboration that Pete Kine and a handful of CDU researchers have had with the Malik Rangers over the last 14 years, which have essentially resulted in the sawfish rescues. And I'm basically going to be summarising the data from these rescues. So today I'm going to be talking about how stranded, critically endangered largetooth sawfish can provide insights into habitat and kinship. Before I go on, I'd like to firstly acknowledge the Larrakia people, traditional custodians of the lands on which we meet today. I'd also like to acknowledge the Malik people as the traditional owners of the land on which all of this work is conducted. I'd like to acknowledge any Aboriginal and Torres Strait Islander people present today. I'd also like to pay my respects to elders past, present and emerging. Now, it shouldn't be a surprise to you that sharks and rays are declining globally. According to the IUCN, approximately 1/3 of all species are currently threatened with extinction. Now, these declines have been driven by overfishing, persecution, climate change, habitat loss, and they've all been compounded by the fact that elasmobranchs banks have slow life histories, meaning that even under low levels of extraction, their populations aren't able to bounce back. Now, an example of this is the family Pristidae, or the sawfishes. All 5 species of sawfish as of last year were listed as critically endangered by the IUCN. And again, their declines have largely been attributed to overfishing due to their commercial value, habitat loss, specifically of mangrove and estuarine coastal habitats, and then their slow life history. So sawfish specifically are case selected, and they really aren't able to experience any population growth under any level of extraction. Now, sawfishes globally have experienced about a 60% range contraction due to these pressures. One of the most evolutionarily distinct species of sawfish is the largetooth sawfish, Pristis. Now, this is a Eurohaline generalist species, essentially meaning that they can use entirely fresh and entirely marine waters across their life stage. They exhibit delayed dispersal from their riverine nurseries, where they will reside until they reach maturity, at which point they'll disperse into coastal habitat. Now, they're large-bodied, they'll reach about 7 metres in total length and live for about 44 years. And as I mentioned before, they're highly threatened. However, whilst they are listed as endangered under the EPBC Act, Australia is still considered a lifeboat population for 4 of the 5 species of sawfish in the world. Not only this, they're culturally important for several groups, not just here in Australia, but also around the world in Central Africa, Asia, South America. They really are quite an awe-inspiring species that attracts a lot of mythology with it. Now, population genetic studies to date suggest that this species exhibits female phylopatry, where females return to the same river system to give birth repeatedly, and that there's male bias dispersal. Essentially, males are free roaming and will mate anywhere and everywhere, and basically they're the main cause of gene flow in this species. This has been demonstrated through matrilineal population structure by Phillips et al. 2011 and Theory et al. 2015, who identified single river basins as the most appropriate management unit for this species. And then this was further consolidated by Nicole Phillips in 2017, who when examining nuclear DNA, saw no population structure across the entirety of Northern Australia. Now, what this means really is that this species is vulnerable to local population declines and extinctions with the limited capacity to recolonise due to low rates of inter-river movements. Now, to take a step back, it's important to know that wet season productivity, wet season drives productivity in Northern Australia. And this is largely associated with that lateral connectivity that we get during flood pulses, where terrestrial resources become inundated, and essentially there's increases in biomass and productivity at all levels of trophic, or at all trophic levels. Now, the monsoonal activity or monsoonal strength has been shown to directly impact the recruitment of commercially important species like barramundi, and also of largetooth sawfish in the Fitzroy River system. Which brings us to what we're going to talk about today. So the first thing I'm going to do is I'm going to overview the Malak sawfish rescues and collaboration. I'll then discuss some insights into the habitat and ecology of juvenile or young of year Pristis that we can get from these records. I'll then discuss some models that we can use to predict both presence and abundance of sawfish on the floodplain. And finally, I'll present some preliminary patterns of genetic relatedness between a handful of individuals caught in the first couple of years of this project. So to characterise the study area, all of this work has taken place on the lower reaches of the Daly River. Now the Daly is a massive catchment, 53,000 square kilometres. It starts up in the western side of the Island Escarpment in the Stone Country, travels through a variety of habitats down where it terminates in Anson Bay, which flows into the Timor Sea. Essentially, like most Northern Australian rivers, the Daly experiences 90% of its rainfall between November and April in the wet season, and then it goes through a dry season, at which point floodwaters generally contract to little pools that look something like this. Now, specifically on the Malak Land Trust, it's a rolling black soil plain for most of the floodplain, with isolated pockets of water like this that can either be grouped into 3 different stages: ephemeral water bodies, semi-permanent water bodies, and deep permanent billabongs. So basically, the focus is going to be on what would be called Group 2, or these semi-permanent billabongs, with an occasional reference to these deeper water holes for comparison. So let's get into the first stage of this, the Malak Ranger collaboration and sawfish rescues. Most of you must be wondering, what are these sawfish rescues? Now, essentially, they're a combination of traditional ecological knowledge and Western science and direct attempt to have a conservation outcome. So basically, this involves locating and surveying for sawfish on the floodplain, catching these sawfish through what are known as the rescue or sawfish rescues, processing these animals, so taking measurements such as total length, the sex of the animal, and then taking a fin clip for downstream genetic analysis, and then relocating them to the main stem of the river, where hopefully they'll grow to a nice old age and make lots more babies and support the population for generations to come. Now, these rescues really came about in, I think it was 2012, when Pete Kine and a handful of CDU researchers were meeting with the Malak Rangers to discuss potential sample locations for sawfish more broadly, not specifically with the rescues in mind. However, when some old ladies from the community were on the land trust hunting for turtles in one of the billabongs, they actually came across, I think it was like 6 or 7 stranded sawfish, and thought to call Pete and the CDU researchers and basically go out and relocate the sawfish. And there the rescues were born, and it's led to a 14-year-long and still it's ongoing collaboration between CDU and the Malak traditional owners and rangers. We've rescued a total of 113 sawfish so far, and the project's been a finalist twice now for the Tanner M Awards Best Collaboration. Now, it's also engaged more broadly 7 different ranger groups, including 5 last year, and it's received global recognition by the likes of National Geographic, the ABC, as a unique conservation initiative, because it really doesn't happen anywhere else in the world in this way. This is like a proactive monitoring and direct conservation intervention. And a real key to this project's success has been the collaboration with the rangers and the importance of traditional ecological knowledge in guiding how we sample, where we sample, and when we sample. And this has really provided a direct conservation benefit. Relocating 113 individuals, even if 10% of those live to maturity, benefit for the Daly River population specifically, especially if those individuals are females, is almost immeasurable, because that's really what you need to support a population. The second important outcome of this is protecting country. Not only protecting the sawfish specifically, but also protecting and working in culturally important ecosystems like the billabongs that have been recognised by the traditional owners as ecosystems of cultural significance and importance for food and for an ongoing connection to country. The rescues have also allowed us to engage in two-way knowledge exchange. We rely on traditional knowledge to do these rescues, and the amount that we've learnt on country from the traditional owners is immense. But it's also provided us the opportunity to contribute the knowledge that we have to the rangers to help them manage the land trust and understand the species in a more holistic or in a more Western science way, understanding patterns of dispersal, like where the adult sawfish actually may go. But in all, the key takeaway here is that the collaboration has been the key to this project's success. Now, what can we actually learn about habitat and ecology from these rescues? Well, the first part of this is that floodplain billabongs provide refugial habitat in the dry season for juvenile large-toothed sawfish. What you see here are the two locations that sawfish have been pulled from over the last 14 years, and all the dots denote different sampling locations really between the two. Now, it's been shown that, well, we've observed that sawfish are specifically using the shaded areas by freshwater mangroves during the day, which may act as thermal refuge because these pools are quite shallow, no deeper than about 1 metre at most. And it's been shown elsewhere, specifically in the Fitzroy, that young of year sawfish actually only use water shallower than 1 metre. And this has been suggested to provide some sort of metabolic benefit, where essentially shallow water is warmer because elasmobranchs rely on ambient temperatures to grow, and young of year sawfish are most vulnerable to predation during this life stage, which may also explain why they're using these billabongs, where predator densities are much lower in this habitat than in deeper, more permanent waterholes, and also the main stem of the Daly River. So if sawfish can experience accelerated growth and reduce predation, this starts to paint a picture as to why they actually might be residing in this habitat. When we look at the length distributions, we can age these individuals all to a 0 to 1 age cohort, which means they all would have been pupped the previous wet season to the year that we were pulling them out. Now, the average total length was about 112 centimetres, plus or minus 7 centimetres, with the biggest individual being 124 and the smallest being about 92. When you put that into the total, the age range, or the age at length range for large-tooth sawfish, it's about 72 to 120 centimetres. So what we see is that these individuals are considerably larger than many of their peers that may not have been able to access these resource-rich and thermally optimal environments. So essentially, they may be experiencing accelerated growth, getting past that critical age length at which they're most vulnerable to predation. And anecdotally, what we find is that this habitat is resource-rich, even into the late dry season when water quality is starting to deteriorate. What you're seeing here are 4 different species of teleos, all of which are likely prey species for juvenile large-tooth sawfish, specifically benthic species like hurdles catfish. Now, I also have a handful of stomach content samples on my desk that I haven't had a chance to work through, but once we can get through that, it'll paint an even clearer picture. All this to say that it seems like we believe that sawfish are exhibiting a trade-off. Essentially, lowland floodplain habitats are resource abundant. That's been demonstrated by a body of work on the Daly River, as well as anecdotally here. It's also been demonstrated that wet season size in the Fitzroy correlates with body condition of juvenile sawfish or large-tooth sawfish. Now, whilst they are using these shallow habitat, they also use deeper water holes at other locations on the floodplain, which have been shown to hold an even higher biomass of fish in papers, I think it was Pusey et al. 2019, 2020. Sorry, that was for you, Colin. But so All of this comes at a cost though. If the wet season rains don't come early enough, then these sawfish will perish. If water quality deteriorates earlier than expected, they will perish. But if they can get out, they're likely in a better body condition at a larger size than their peers, and therefore are able to avoid predation in stream better than other individuals from the same cohort. Moving on, now I'm just going to quickly run through some models of presence and abundance. Now, the first model was simply looking at what wet season characteristics can we use to predict sawfish presence in a given year. To do this, I used a logistic regression with a complementary log-log link. The second model was essentially the same thing, but with abundance. And to do this, we used a zero-inflated negative binomial. In both instances, to select the best model, delta AICC was used. Now, from our first model, we can say that the average wet season river level shows a significant positive correlation between, there's a significant positive correlation between average wet season river level and abundance, with a single unit increase in average wet season river level, increasing the odds of sawfish being present on the floodplain by about 2.5 times. From discussions with the rangers, the floodplain actually becomes inundated at about 4.5 to 5 metres. And what we can see is that if the wet season water levels are at that 4.5 metre level, then the floodplain is likely inundated for the entirety of the wet season, or almost the entirety of the wet season, at which point sawfish have an opportunity to disperse onto the floodplain and access these resources. A similar pattern is observed for abundance, where there's a strong positive relationship between AMI and abundance. However, we have few data points, and that's worth noting for both models, but we still see quite a strong relationship where as the AMI increases, the abundance or the predicted abundance of sawfish in a given rescue year increases quite significantly. All this to say, presence and abundance are strongly correlated with wet season strength, i.e. the AMI, and the extent of floodplain inundation, or the average wet season river level. Finally, I'm quickly going to run through some preliminary relatedness analysis. Now, relatedness is just the genetic similarity shared from a common ancestor. Basically, our analysis used 4,700 SNPs, 29 individuals sampled in 2012 and 2013, and a multi-estimator approach. If you want to know more, you can talk to me about it afterwards. We tested for non-random grouping of kin spatially and temporally. So to do this, I used a generalized linear mixed model where individual 1 and individual 2 in each pair were random effects, and that just accounts for the non-independence of a lot of the observations. So basically, interpreting results here, if you've got lots of siblings in your data and they're non-random, that means you've got a tiny population and they're on pretty dire straits. If it's non-random, then you're likely observing fine-scale kin structure or some sort of kin selection behaviour as two potential explanations. Now, our relatedness analysis showed that there were 5 different sibling groups in the 29 individuals, with 6 full sibling pairs and 4 half sibling pairs. Now, on this plot, I've just pulled out the related individuals. Any unrelated individual won't be visualised here, but basically blue lines denote full siblings, orange lines denote half siblings, dotted lines denote individuals that were caught in different locations, and solid lines are individuals caught in the same place at the same time. And essentially what we see is that 6 individuals were caught in the same location and 4 were caught in different capture locations. From the GLMM, what we found is that the odds of being related increased by 3.25 times when caught in the same location. However, this result was non-significant. There is still a very strong effect, especially when considering the fact that there's a low baseline probability of being related with the other individual when you're not caught in the same location and you're not caught in the same cohort. Now, there's also a lower odds of being related when caught in the same cohort, and there's a strong and significant individual level effect in our model. So really, what we've found is that there's a low incidence of kin, with only 2.5% of all of the individuals observed in the data set being related. And that we found that capture location may affect the odds of being related. However, we obviously need more data, which we have, to refine that relationship. It's important to highlight that all of this conservation work is underscored by the fact that the daily is under threat by the likes of agriculture, water allocation, and climate change. Now, irrigated agriculture and potential floodplain harvesting is going to wreak havoc on the natural flow regime of the systems like this, where essentially through this water allocation and pumping, the floodplain will be inundated potentially for a shorter period of time, which reduces the opportunities for all species, not just sawfish, to disperse onto the floodplain and access these resources. It may limit the ability to recruit for many species, and it also limits their ability to disperse back into the river, which increases the likelihood that they may be stranded somewhere that's unfavourable and end up perishing well and truly, or much earlier into the dry season than they otherwise would. The second risk factor is climate change, where essentially an increasing gap between large wet seasons may limit the opportunities for sawfish to recruit more generally, and it may decrease the productivity of these systems overall. To wrap it up, some 4 key takeaways for you all. So the sawfish rescues are essentially conservation and collaboration, incorporating traditional ecological knowledge to have a direct conservation outcome. Now, the second point is sawfish use the floodplain habitat as refugia to increase growth and avoid predation, and they're exhibiting a trade-off where the potential risk is that they will perish. We also have observed that presence and abundance correlates well with average wet season river level and monsoon strength, and this highlights the importance of natural flow regimes and climate action in the present day. Now, and finally, that overall relatedness was low, but that individuals caught in the same location increase the odds of being related. And our total data set has 151 individuals that will be genotyped, and that's really what Martin is going to focus on. But yeah, thank you.
Morning, everyone. Thank you very much for being here so early. And I'm going to present part of my chapter 2 for my thesis. So it's related to Asia region. So first of all, I would like to acknowledge that there are Araucarian people of the traditional custodians of the lands on which we live today. So as I told before, this is part of my chapter 2, and my thesis title is Identifying and Monitoring Critical Shark and Ray Habitat in the West Pacific through Citizen Science. So this is going to be based in the Asia region for sharks and rays critical habitat. So as a short introduction, elasmobile branches that are sharks and rays are a very diverse and ancient group of cartilaginous species that include 1,300 species. So it's very diverse, and there are key species as top predators and mesopredators. And they are the second most threatened vertebrate group worldwide. So they are after amphibians. So there's a lot of concern towards their conservation, and overfishing is their main threat, as it is for most species in the marine world. So Asia is a very important region for sharks and rays, and globally it holds the epicenter of marine biodiversity. It has a high habitat complexity, and it has a very high species diversity for sharks and rays. So around 31% of all sharks and rays are found in this region. It also has a high range restricted species, where we find 30% of all species in this area are restricted. And it has a high functional richness, which means in this area sharks perform different ecological functions. But this area has a lot of threats. So Asia is a major player in the global capture and trade of sharks and rays worldwide. It has leading fishing nations, Indonesia leading, followed by India, China, Japan, and Malaysia. It's a trade hub, especially in the Southeast Pacific, for multiple parts like fins, skin, meat. And catches are declining, so this is an indication that the populations are overfished. And this area has higher than overall threatened species. So almost 50% of all species in the region are threatened with extinction. And there has been recorded species extinctions. For example, one of the most iconic that recently was declared extinct was the Jabari ray. So this species is gone, and it used to be located in Indonesia. So it's a region that holds a very important ecological but otherwise has a lot of threats that need to be taken into account. So what is the problem when we want to conserve sharks and rays? Well, conservation of sharks of this group is challenging due to their high species diversity and complex life history. We have species through all types of habitats in the ocean, even estuaries, and even inland waters. So in this case, and one-size-fits-all approach is ineffective and has been proven that it doesn't work for this group specifically. So effective conservation needs to consider the species-specific ecological and biological data. So protecting their habitat, it's a crucial way to protect. So that's where the important shark area has appeared as a way to try to solve these gaps in knowledge and gaps in management. So this is run by the IUCN Shark Specialist Group, and it's a new project that started in 2021. So far has a map, the critical habitat of half of the world, and it's an approach introduced to identify and protect critical habitat. So critical habitat basically is any 3D spatial area that is critical for the life history of sharks and rays. So the ultimate goal of this project is that it will provide decision makers with actionable knowledge. So there is a website that everyone can access, and we have an e-atlas with all the areas that you can download the fact sheets and the maps. So the aim specifically of this project is to assess the critical habitats identified for sharks and rays through the ISRA process in the Asia region. I'm going to present the four most important objectives. The first one was to identify which species are eliminated through the ISRA process, what are extinction risks, habitat types, and the ISRA criteria that was used. I'm going to talk about that later. Then assess whether these ISRAs overlap with hotspots for biodiversity, range-restricted species, and functional regions that is related to the ecological functions they provide. And then evaluate which data methods were used to identify those critical habitats. And finally, assess the overlap of the ISRAs and marine protected areas in the region. And finally, will be to identify what are the research gaps and provide recommendations to increase the conservation of this. So to identify the ISRAs, we work region by region, and we dedicate around 6 months collecting all the information that is available that could be in forms of publication, reports, local ecological knowledge, city science, whatever it's out there. And we also gather in January 2024 with all the regional experts here. So we invite everyone that is involved in critical habitat in the region, and we map this critical habitat. So the process does go through peer review. It's the same when you submit a paper on a journal. So we have an independent review panel that is run by Colin Simplendorfer and a group of renowned shark scientists that evaluate these areas if they apply for the criteria. I'm just going to show how it works. So in total, 122 ISRAs were identified in this region. So this is the final map for this region that I'm going to show you, and we're going to go through it in more detail. So there are 4 criteria that are used that are subdivided in sub-criteria. So vulnerability, it's very easy. It's basically if the species is threatened or not, it will qualify for this criteria. But this criteria needs to be, comes with another criteria. That cannot be by itself. So range restricted is basically any species that is located in 1 or maximum 2 large marine ecosystems to qualify as a range restricted. And then the most comprehensive one are the criteria C, that is life history. So the first one is C1, that is reproductive areas. So these areas are important for mating, for nursery areas as well, where you find pregnant females are also egg case. Egg case nursery areas. And then feeding areas are areas where they are important for feeding processes, but they need to be exceptionally important because if not, everywhere in the world will be a feeding area. So then will be places where the resources are concentrated. Then we have resting areas where shark rest. And then we have movement areas. Movement areas usually are very big areas because they connect different life history stages. For example, shark migrates from a feeding area to a reproductive area and vice versa. And then we have undefined aggregations where sharks, we know that they aggregate regular and predictable, but we don't know why they are there. We know there are huge amounts, but still there's not enough knowledge, so they don't use type of aggregations. The group is very big. And then we have a criterion that is special attributes that is subdivided in distinctiveness. That, for example, this is a very cool case in Galapagos Islands where there's scientists studying these white skates. They use a nursery area, the deep hydrothermal vents because the temperature is a little bit higher. So that's very unique because other species have been identified doing that. In this region, one of the areas I think was a cleaning station for treasure sharks, and there are very few worldwide. So that will qualify as distinctiveness. And the diversity is basically if the area has a high diversity of species. For this region, we have chosen a threshold of 31 species. So in each region, it changes depending on the total marine species diversity. So for each ISRA, the information that we collected was qualifying species, they're the extinction risk, the ISRA criteria that they qualify for, the habitat type, and the method that was used to collect this data. So this could be very varied from aerial surveys, we have independent fishery surveys, we have citizen science, we have under remote cameras, satellite data, and as well a landing or fishery information. And there are much more methods being used, but I'm going to focus on the most important. Then we identify the hotspots in the Asia region for the biodiversity, for functional regions that are related to the ecological role sharks perform in this area, and also for range-restricted species regions. Once we identify all these areas, we overlap with the ISRAs for all these three, and then with the marine protected areas. So the results were that 122 ISRAs were identified in this region, and this is all around 1 million kilometers square. So it's a huge area. It still only represents 3% of all the Asia region. So the Asia region is the black marks you can see over there. So it is a huge area that goes all the way from Russia to Indonesia. The majority of these islands were located in national waters, so only a few were international, and we have, I think, 6 were transboundary that were shared between countries. If we look at the jurisdiction level, we can see that Indonesia leads with not only the numbers, there are 41 ISRAs, but also with the size, total size that they accumulated. So this ISRA represents 30% of its water and also 72% of the total ISRA area. But as you can see, most of these areas were movement areas. So movement areas are huge areas that are basically, well, I'm going to tell you which is the species responsible for these movement areas. But if we discard or exclude movement areas from the analysis, we can see that Indonesia still is the most important country for the ISRAs identified. And that it's 21% of total ISRAs in the region. And this is followed by Russia, that has only 7 lists, but the sizes they have is larger. And it has India, and some like Cambodia, and even a few countries we couldn't identify in ISRAs, unfortunately. Timor-Leste, South Korea, there was no ISRA. No experts in racial Korean, no information. When we go to the species level, we can see there are 121 species that were identified in this region. We have more rays, which is weird because other areas start from the one who were taking off. We have 53 species of shark and 77 species of ray and 1 chimera. Most families that we identify were Calcarinides with 16 species. Then we have Mobules with 7 species, Daciatides 20 species, and then Spirnides with 3 species. So these are the 4 families that are most representative in the ISRAs that have been identified in this region. If we see at the species level, we can see here the whale shark is the most representative species in terms of size. If you can see here on the x-axis, you see that this is a size, total size area that whale sharks occupy. So that's the most representative species, and it's followed by the scalloped hammerhead, where it has small ISRAs in size, but a lot of them. And then we have black-tip reef sharks, reef manta rays, oceanic mantas, bottlenose whitefish, and bull sharks. Those will be the 6 species that represent 81% of the ISRA are included. So as you can see, there's a bias because these species have greatly studied and we were able to identify their critical habitat, while 65 species that represent almost 50% of all the qualifying species identified only have 1 ISRA. And still, 70% of all the species in the region have no ISRA identified. Although we have a lot of information, there's still great gaps of knowledge. The good news is that 76% of these species that we identified are threatened. It's good news because we have been, all these studies have shown that we are focusing our research effort on these species that are endangered, threatened. And most of them are. And for the ISRA criteria used, we can see that the most commonly used criteria is vulnerability. But life history then was the second most important criteria used. And other aspects like species attributes have very low ISRA. So for the life history, we can see our reproductive areas are the most important, especially for nursery areas like newborns, young of the year, and pregnant females. And then we have also feeding areas that are the second most important. So if we want to look at how they are distributed in the 3D habitats, I'm not going to go in detail in this graph, but basically most of the ISRAs are located near shore in the coastal region on the superficial layer. So most of them are located in the first 200 meters of depth and very restricted to the coast. This doesn't mean that there's other, just doesn't mean that all the research effort is focused in that region. And then I'm going to go through the hotspots. So I'm going to start with the biodiversity hotspot for sharks and ray. So 21% of ISRAs show high overlap with areas of high biodiversity, as you can see in this map. And 3 areas exhibited exceptionally high biodiversity. This is the Taiwan Strait here, where we could identify 2 ISRAs. Then we have Polk Strait that is located in Sri Lanka and India, with 2 ISRAs identified. And then we have the Sarawak State in Malaysia, where 1 ISRA could be identified. So there's some overlap, but there's still a lot of work that needs to be done. For the rate-restricted, we find that only 9% of ISRAs overlap with areas of high restricted species richness. When we look to the areas that have the exceptionally high restricted species richness, we found that central Japan, central southern Japan in the Pacific side was an important area for restricted species. Then it was followed by Taiwan and Polk Strait again. So we see some patterns shared with the biodiversity. And then finally, where it's important areas where there's a high richness for functional ecological race. So we found here that, 88%, that's a good high overlap with ISRAs with these high areas. And we only identified 2 areas with exceptionally high functional richness. Again, Taiwan was one of the key areas, and this was followed by South Central Japan in the Pacific Ocean. So the methods used. Which was the method that was most important for identifying critical habitat? Well, it was a surprise. The citizen science was the most important, and this is basically by divers. So we know that this area is important for the diving industry, and they are doing a good job providing information to this critical habitat. It was followed by fisheries data, mainly gill nets, as a direct use was the most important, and this was followed by visual census, ecological knowledge, and market knowledge. I'm going to go briefly because this is how methods were used according to each jurisdiction. So it varied a lot across each jurisdiction, but you can see, for example, that Indonesia was one place that had multiple different methods, including citizen science and as well fisheries. Then the Russian Federation, for example, only basically used scientific surveys, but they were focused in deep water scale species, which was unique in this region. And then others like Philippines, and also like Philippines, they use basically remote video. So this is interesting because jurisdiction can make collaborations between them in the future, sharing their unique type of methods used. And when we check how the methods change by the criteria, we can see, for example, that citizen science is used in different types of criteria. Like we know that life history is mostly used, except C4. So C4 is movement. So movement is basically electronic tracking, of course. That makes sense. It's the one that is most used for this. And then we have like commercial fisheries is used for feeding areas and reproductive areas. So each method basically is looking for different aspects of the life history of sharks. When we look for marine protected areas, there's 1,343 MPAs in total, and predominantly they all are partial. So we did differentiate MPAs for no-take that fishery is totally banned and partial areas where fishing is allowed. So in numbers per commercial MPAs are the ones who dominate, but when we look at sizes, then it's no-take that have the largest. But this is mainly represented by 6 big MPAs that are from Japan and Indonesia. And only 5% of these MPAs overlap with ISRA. So that's a low. So this is how each jurisdiction has the total MPAs in their exclusive economic zone, that's its waters. So you can see that Japan has the largest in terms of size, followed by Bangladesh. But interestingly, in this region, no country reached a target of 10% protection and 30% protection. So internationally, there has been an agreement between countries to protect this water, and none of them, and the 10% target was due in 2020, and the 30% is 2030. So they are very low, behind. And many countries protected less than 1% of their waters, like Taiwan, Timor-Leste, Vietnam, Myanmar, Russia, Sri Lanka, Brunei, and Singapore. Those are the countries that have the less representative case. So when we look at the spatial overlap, we can see that 40% of ISRAs overlap with MPAs. And MPAs overlap it's mainly with reproductive areas and defined aggregations in feeding areas. And 72% of species that represent 39% of the total species identified overlap with MPAs. But again, most of these species are basically whale sharks, scallop hammerheads, reef manta rays, and black. In terms of jurisdiction, you can see that Thailand, followed by Bangladesh, are the countries who have the highest overlap with ISRAs, with MPAs. So key findings, finalizing, is that Indonesia stands out with the highest number of ISRAs and the largest accumulative area, which means that most of the research effort has been focused in Indonesia, which makes sense because it's a huge exclusive economic zone and it's one of the high biodiversity regions. Most ISRAs were reproductive areas and were located on the coastal environment with specific habitat attributes. We were able to identify 121 shark and ray species that represent 30% of the region's diversity. Yet most ISRAs, both in number and size, are concentrated around a few species. Most of them are charismatic species with wide distributions like the whale sharks and the manta rays. And they're very important for tourist industry. While others like range-restricted species and deep water species are poorly represented, which demonstrates that there is a great gaps of knowledge for these groups. The good thing is one of the most studied species, that wetfish, is poorly studied, and this group of species are also very critically endangered, most of them. I'm good at time? No. Okay. So some ISRAs show a high overlap with some of the exceptional shark and ray biodiversity, range richness, and functional richness. However, the degree of overlap varies a lot. So this could provide recommendations to inform target management. Importantly, that citizen science stands are like a very important method for delineating ISRAs. Although citizen science is focused in this high biodiversity jurisdiction, we also need to consider that it's very focused in charismatic species and also in a specific that like coral reefs. Fisheries was the second most important, but it could be used more widely in this region, considering that this is one of the most important fishery regions in the world. For example, this species of shark, Carinosaura, it's one of the most important commercial species in the region, and we could only identify one ISRA. And there are 38 highly commercial species in this region, and we couldn't identify any ISRA. For fisheries, it's a strong method that should be used more widely in this region. And finally, only a small fraction, 5.4% of ISRAs overlap with MPAs. So this region still falls short behind meeting the targets for protection. Then I would like to thank the ISRA team, the IUCN has been supporting this, and Shark Conservation has provided the funding for this. And I would like to invite everyone to check our website. You can have a lot of information. We have resources, we have publications, and this is the e-atlas map that you can access. And Australia is coming as the next region that we're going to assess. That is going to happen around August, September, and there is a chance it could happen here in Darwin. So thank you very much for your time.
Yeah, and stay here and have a question. Would you guys both like to come back up for questions? All right, any questions in the room? Right, so I've got a few. Go for it.
As in where they're pupped. We don't know for certain, but the general consensus, or I guess general consensus among people is that they get pupped either at the mouth of the rivers, and then they swim upstream. So during like the first freshwater pulse that comes through the rivers, essentially that may trigger pupping from the females. But yeah, we don't know for certain.
No research is like more worldwide in other areas.
It's like, yeah.
Question about, with you mentioned that there was ones that were relocated 100 something, and you said it'd be nice if they were females. I was wondering how many were female. And I guess the question was you'd mentioned about the what I think is you're talking about the thermal environment in those pools, but then on the other hand you were saying, what I gather you were saying it's better if it's warmer, but then you were saying they were sitting in the cooler areas. So I'm wondering how that is then helping their physiology. And the other thing was with the freshwater mangrove, which I think you mentioned there. I was wondering what you mean by freshwater mangrove, because I assume you mean Barringtonia, but you didn't have the species.
Yeah, that was that was the species. But yeah, I can talk through that. I actually have a slide, I'm pretty sure, that has the sex ratios. I was going to talk about it, but in the interest of time, cut it out. Extra stuff. Spinning wheel of doom. Yeah. Right. No, maybe did I cut it out entirely? Oh well, never mind. That was a great waste of time. I promise I'm not filibustering. No, basically, sex ratio was about 50-50. I think it was about one male for every 0.8 females across all of the years. In 2024, so last year, there was a real skew towards males. It was about 3 males for every female. But obviously, that's a single data point. But if that continues, then that's alarm bells from a replenishment perspective. In terms of the thermal side of things, obviously there is a point at which things aren't thermally optimal. So, and those pools only about 600 mil deep, and that water on a 35 to like 37 degree day, that's going to get really, really toasty. And when you've got anoxic conditions as well. All right, yeah, we do. We have environmental data. I have it handy when I think it didn't get shared with me before the presentation, so we've included it. Yeah, we do have temperature. And then, sorry.
Thank you both for your presentations. I just had a question for Sam. With it's a lot of species use these floodplain environments, but it's this precarious environment where there are clear benefits, obviously, of why they're going there. But then at some point, like you're saying, both in terms of temperature and habitat availability and other environmental changes, these animals need to leave the environments. And is there any evidence with sawfish that these juveniles, if the conditions are right, will they stay on these floodplains, in these wetlands for multiple years, or is it thought to be that it's the young of year that move onto the floodplain and then they're trying to get off, and these are every year? Do you have that kind of information, or have other people studied that?
It hasn't been studied, but we haven't recaptured any individuals in the 14 years this has happened. But the sawfish reliably occur in the same places. So obviously we're removing the individuals, so unless they were to disperse back, get recaptures. I'm not entirely sure of the total lengths of individuals that are in the deeper pools, but maybe larger individuals are using, like to throw an idea out there, maybe larger individuals are using habitat that has a higher density of predators because they're less susceptible to predation. But yeah, in terms of young of year sawfish, they're the ones that we're finding, and then the only large individuals are in the main stem of the river.
Yeah, right.
It's quite interesting.
Have you sampled the permanent deeper wetlands?
Yeah.
Because I know Steve Purcell, not Steve, forget his name. He was in Queensland. He left. There's this guy, but they Pebble, and they found populations in deep permanent wetlands, the Flinders Norman kind of complex. But the other thing I was going to ask is, and this coming from the fish point of view, as far as that pupping thing, can you use their teeth on their rostrum to stable isotope where sort of the sulfur, I guess it would be the sulfur, range from when they were born versus when they get into the salt, and then sort of link that to water quality within the river to determine where they are actually pupping?
I'm not sure. I'm not a stable isotopes person, but like
Has anybody done that with them?
I know someone who has used. I don't know if they've used. I think they've used isotopes. Nikki has used stable isotopes, so to basically do exactly that. But that's more for like trophy saws to kind of identify where they were from.
Yeah, because you can use laser ablated stable isotopes on otoliths to actually do this isotopes for dating where they are trying to indicate where they come from rather than trophic.
Oh, if you doing it, I can. I think
Just
this.
just a question. I didn't talk to you.
Oh, all right. Thank you. It was very, very good presentation. Thank you so much. I have a question for Adriana. Back in the day when MPAs were set, they were more thinking of a umbrella protection to everything that happens over there. We surely know that species under fisheries do have a better performance in MPA's areas, whether they are no-take or even open to fisheries. In this particular case with the ISRAs, they seem to be very species specific. And they seem to be a very good way to identify on a species level. Would you decide to move into something like a umbrella ISRA that will attempt to promote conservation of a bigger number of species? Because then I think the governments will be much more interested in setting up ISRAs because they think, oh, we just set one ISRA and we're going to report that we have provided shelter to 35-7 species. You know, they make up. So is that something that you will be thinking about, how to move from species-specific ISRAs to something that is a bit more like a blanket coverage for endangered species in the Asian region?
That would be optimal, no? But sharks are very, because they're very important for the ecosystem, they could function as indicators, no? And for ecological and abundance rates, for example. We did an analysis for MPAs in the Eastern Pacific where we identified that very few MPAs use sharks as objective species. So I don't say that they're the most important, but they have been very disregarded historically as important species. In this region, we are not going to go to that level of analysis because there are much more MPAs and every country their own language, so it would be like a headache. But it would be something interesting to identify which ones have sharks and rays as objectives for conservation for each MPA. My guess would be like worldwide, no, most MPAs are not designed for sharks. So we need to balance that out. Most are very related to more charismatic species like marine mammals, sea turtles, which is great, but we need to open our scope for other species.
I have a question. So you mentioned that citizen science and fisheries were like where you got most of your information from. Are there any other methods that you want to see more information come from? And is there a way you can like kind of build on that, like those methods?
Yes, but I think citizens, first, my personal opinion is citizen science has a great value because it's basically almost for free. No, like you can gather, especially in this region where it's one of the most dived areas in the world. And a lot of people come just to see sharks. But it's almost focused on a few species. So if that could change, for example, for range-restricted species, like wobegons, like where they have high diversity, they're very shallow species, that would make a huge difference to study more. and fisheries as well. This is the nation that hosts the highest fishing in the world, and sharks and rays are part of that. And as I have seen, viewing the IUCN report that I was released last year, there are 40 species that are highly commercial. We don't have any ISRA. And they are very low cost. You need to consider as well the cost of that. For example, tracking, it's incredibly expensive, and it's basically done on bull sharks in huge areas that are going to be impossible to protect. They're still important, but we need to focus on the social aspects in this region. But yeah, like for example, remote video was not that used. Philippines was used, especially in very remote locations like MPS that are in the middle of nowhere. So there are a bunch of aerial surveys are rarely used. I think very few areas that will also be increased. So I think it will be good a collaboration between jurisdictions to share their own type of.
Yes, I have a question for Sam, and Adria might need your help on this one. I read about Pristis in the Amazon basin. Am I right? Okay. So has there anything like that been reported in the Amazon? Because the Amazon, the lower lands of the Amazon is also a floodplain, and there's lots of indigenous people living over there. Are there also strandings of yearlings in there? That would be an amazing.
Actually, one region that we are just finalizing assessing is inland waters. I have been greatly involved and learn a lot of inland species. And sawfish is a species that is rarely seen. Like we still have a few areas, estuaries areas, but it's just one record on the last 5-10 years. It's more traditional ecological knowledge. They hold information. So it has been hard because sometimes we need pictures and sometimes they say they see them, but we don't know what species exactly it is. So that is coming soon.
Outside of Australia, sawfish are pretty much stopped. Like they're not really in the densities that, and like sightings like that. Yeah.
I remember the paper about life rafts.
Yeah.
Quick question for you both. Mine actually, Sam, relates to this one.
Yes.
You mentioned that obviously there's potential pairs or groups. I guess my first question is, and then there's half siblings, which could potentially be the same year. Can mothers have multiple partners for the offspring? So if they're having multiple times for the same litter group, because then potentially you could be having, even though they're half siblings, there could be litter behavior where they're hanging out in the same groups, and same for your full siblings. So my question is there a way to work out when they're born, so year of birth, and then look back at genetics to see if it actually is family patterns, if there's groupings, or if it is just random?
Yep. I mean, like that's kind of what we tried to do here is like, so we know when they're born because they are all young of years, so that they would have all been born, and the year that we sample, they were born the wet season prior. So I mean, you could lump them into cohorts, so it's like wet season 23, 24, for example. And then
because growth you mentioned is varied due to temperature, you can have something grow faster. How do you actually know that they're born at the same time if their growth is so varied?
Because they're not going to, so sawfish, it's a fair point that they may be like a small individual that is 2 years old. But I mean, those total length ranges are pretty well established. Like sawfish have quite a lot of life history for an elasmobranch, like life history knowledge. So I would say like we have fair confidence that between that 72 to 120 centimeter total length range, they're young, so 0 to 1 years old. But yeah, like odds on these guys are all closer to that 1 year age mark, at least like 7 months.
And Adriana, my question for you was obviously the shark and rays diversity is like understudied for deep ocean. Do you think it's important to be trying to, obviously it's just not your survey, but everyone's surveys are biased towards shallower depths, hence why we have more information. Is it important to extend that to deeper areas, and if so, how?
Yes, of course. And that was one of the recommendations of the data that we have, that it was very focused on nearshore environments, so we know there's a lot of deep water species that are not studied, especially in the tropical areas, because, for example, Russia has been done a great job with their independent fishery surveys, and it's regions that is why it's not done. One is because of the high cost to get there, but they have been doing independent fishery surveys. Could be a way to do it. I don't think there's any commercial fisheries in that region or in that level of the ocean, but also remote cameras have been working great. I know there are a lot of studies done by Pristis Nat Geo. They're using crabs deployments, and they are, but they only have one time, and that's it. So we need to do it more regular and predict that more than 1 year.